Getting my jokes.
No.
All right, so we have, starting here, in the way we're going to study this, the way your notes are, we're starting from where the sperm is made to where it travels, where it picks up the rest of the semen until it gets ejected from the male body. Okay, so we have to start here, and this is the scrotum. Okay, scrotum means pouch in Latin. Okay, so this is the housing of the testes. And we have two muscles that we want to talk about. So this one here is the easiest one. It's the cremaster muscle. Okay, then the dartos, you can barely see it. It's just this little rim. Okay, so if you look here, there's this little muscle-colored ribbon right there. That is the dartos muscles. Okay, so we have wrinkling, then we have lifting. Okay, and we want to do that in the male because sperm production is temperature dependent. Okay, so there you go. Okay, and then we have two coverings. And the outer covering is the tunica vaginalis. And then the one that's on the testes itself is the tunica albuginea. Okay, so just in your notes, on testes. Cool? And then if you learn that one and you learn it real good and I tag the other one, then it's like, oh, not the albuginea. But you can't put not the albuginea. You have to put what it is, vaginal. Cool? All right, so this is on the slide. Okay, so let's pop that out. Okay, so the section we have is not this close of a section. You can actually see the full circle, okay, the full tubule, okay, but you're going to see these guys, and you're going to see these guys, and you're going to see these little guys right in here, okay. So if they're hugging, and this is the capsule, okay. So this is outside the capsule. Then you'll have another one of these guys coming over here. So there's a little bit of space between there, okay. All right, what do you notice about these guys that's different from all these guys? You should notice round versus oblong. Okay, so we're looking for an oblong cell. That's your Sertoli cell. This is your helper. This is your big brother. This is the one that guides and helps mature these spermies. It also makes the blood-testes barrier, okay, because we don't want our blood, men's blood, to go in there because they will kill the haploid, the haploid cells that are in there, okay? So here's your spermatogonia, okay? And these do travel, whereas the female in the egg, all those changes that we talked about happened right there at that section, okay? But these do travel up until they get to the lumen. Okay, so here's your spermatogonia. So on the slide, you'll be looking for the border and anything closest to it, okay? Then the primary spermatocyte is all speckled. And they're big, okay? And then the spermatid is round, much smaller, not speckled. It's what we call pycnotic. It's very dark. And I couldn't find any spermatozoa, so I'll keep looking. But if they start looking like the sperm shape, then there's the spermatozoa. Cool? All right. If you're not within that, and you're in the interstitial, then you are a Leydig cell. Okay, this is called an interstitial cell, but Leydig is the dude that they named it after, okay? So this is our border right here. There's your primary, okay? There's the spermatogonia, there's the primary, and then it moves up. This is outside of the tubules, and you can see it very clearly over there in our slide that it's not within the tubules because we have tubule here, then we have a tubule here, and then we have cells, and that's where you're gonna find your Leydig cells, okay? So during arousal, this is where the luteinizing hormone is gonna stimulate the testes, this guy to release testosterone. Pretty cool, huh? All right, so there cells and the Sertoli cells. And, oh, I hope I brought it. We have to use a picture. Oh, I do. This is on your slide presentation, okay? So this is what I have to use, a colored version of this, okay, for this kind of stuff here. Okay, so this is a cross section. I mean, it's over there, but it's so small, there's nothing I can really tag. So this is what I have to do, okay? So there's your seminiferous tubules, there's your lobules, okay, putting these up. The tabulus rectus are these coming from the seminiferous tubules to this center area. The center area is called the reedy testes. So these coming in are the tabulus rectus. Okay. Then the efferent are the ones coming out. Remember, this is the epididymis. Okay, and the epididymis is a big structure that we can see. Okay, so on all our models, we can see this right here. So that's the epididymis. Okay, so that's the epididymis. There's the epididymis. You got another one? Oh, look at this cute epididymis. And if you get its brother on the other testes, it's called epididymi. Isn't that cool? This is Latin. Okay. All right, so this picture is on your slide presentation with the labels, and I will use this to tag this particular question. Cool? Okay, so from the testes, from the epididymis, because the epididymis has a head, body, and tail. Okay, so the head, body, and tail. Then it becomes the ductus deferens. And who's got a better one? This guy has a better one. No, you don't. I don't like that one. I like the back side of this one, but I don't like it coming up from the testes. See, here you can see it from the testes coming up. Okay, so it's going to go around the pubic bone. It's going to around the ligament. So this is pubic bone right here. Okay, then it's going to double back behind. It's going to go behind the bladder. Okay, so the seminal vesicle is number 5 on this model. Okay, this is the ampulla. Okay, or the ampulla where the two meet. So the seminal vesicle is this. It's also better shown with this one here. Okay, this is a gross section. This is a dissected section. Okay, so you can see that this is coming back. There's the ductus deferens coming in right here. This is the seminal vesicle. Okay, seminal vesicle is going to make 60% of the semen. Okay, and since those little spermies have to swim upstream, they need a lot of energy, so they're going to have fructose for ATP production. Okay, because they got a lot of mitochondria. Okay, so that's what's in the mid piece of the sperm. So they need that. Okay, so this is part of it. It's got a clotting agent, prostaglandin. Okay, then the ampulla is where the two meet. And who's got the best version of that? I thought I saw one where. Do I have all the models here that I'm supposed to have? Who's got the best? Oh, you must have the best one. No, this isn't too bad. Okay, so there you can see the seminal vesicle. There's the ductus deferens. Where they unite, they form the ampulla. And then from the ampulla, they go into the prostate gland. This is the ejaculatory duct right here. Okay, it's going to go into the... Well, this guy's not too bad. Yeah, so there's the ampulla. Number 6 is the ejaculatory duct. Okay, now we're in the prostate gland itself. Okay, so prostate gland is going to make 30% of the semen. So we got, again, more stuff for the ATP production. We have proteolytic enzymes to break the clotting proteins that came in with the seminal vesicle fluid. Okay. Then again, just like there's a prostatic urethra, this right here is supposed to be the membrane. So this is the membrane urethra, then the penile urethra. Okay. But we can't just let the sperm out right now. We have to do something, because the urethra in the male has a dual function. The male urethra carries urine, and it also has to carry semen. Okay, well, we know that the acid in the urine is going to kill all those little spermies babies, and we can't have that, right? So we have to have a mechanism to cleanse that out. And who has this? Oh, this one's a good one right here. And let's see, do you have one? No, you don't. Do you have one? Yes, you do. Before we exit, okay, so there's, you can see there's the penile urethra right here. We have this little bulb right there, and that has a number. What's that number?
21.
21. Okay, that's not the name of it. The name of it is the bulbourethral gland, not 21. Cool? It does 5% of semen, but it's alkaline secretion that cleans out the urethra and also produces mucus to lubricate the urethra here. Okay, so that's a pretty important job. It's 5% of the semen. So actually that comes out first, okay? And then you have the rest of the guys shooting out there. Cool? And on this model, it is right there, number 8. That doesn't look like much, but that's what that's supposed to be, the bulbourethral gland. All right. All right, in the penis itself, we already talked about the penile urethra, but we have all this glandular tissue. This right here is the corpora cavernosus, and it's a paired dorsal structure. So if you look at this model, you can see that it's paired structures, okay, and they're superiorly located or dorsally located on the penis, okay. And then corpora spongiosus is the bottom one that's around the urethra. So we have corpora cavernosus and bulbospongiosus, okay? And very necessary, because with all the blood that enters the penis to give it an erection, it could squeeze the urethra. Well, that defeats the purpose, right? Then it'd backflow and stuff like that. And we learned from the other lecture that this also is going to close off, so when this thing comes through, it doesn't backflow into the bladder. Because then that doesn't work either. I mean, if the goal is to impregnate the woman, well, you got to get the sperm into the woman, and that ain't going to happen if it goes up into the bladder. Okay, and then we have the glans penis, and then the prepuce is the skin fold that's over it. Okay, and we talked about a circumcised penis will have a corona there. But I don't think our models, well, yeah, this one did. Okay. I'm not going to get too emotional about it. Okay, and then the sperm, we have the head, the midpiece, and the tail. Okay, the head has the nucleus, and it has the acrosome so that we can burrow into the into the egg. Okay, and we talked about how the female will produce some enzymes that will let that do that and come in there. And then we have the midpiece that is contains mitochondria, hence that's why we needed all the sugars from the semen to do that. And then, of course, the tail is what's going to have to propel this thing in the female tract. And he's got an arduous journey. All right, so now let's look at the oh mama. And the way I've got my notes set out is we're learning the external stuff up until the vulvar area. Then we're going to go from where the egg the egg comes from. Okay, so all the structures the penis has to go through and then what the sperm has to drive through, and then what the egg does in its travels when it comes back. Okay, so these are our mamas. Well, actually, they're not mamas yet. But if we keep putting those models next to them, they're going to be mamas before they know it. Oh, we didn't talk about the muscles here. Bulbospongiosis, ischiocavernosus. Okay, so these are going to help as well as with the pelvic floor and the muscles that come here. So during ejaculation, all those muscles are going to contract and help propel the semen. All right, where's my lovely woman? No men allowed. All right, what am I need one more. Look at all these things. All right, so we're talking about the external anatomy. The external anatomy is this area right in here. It is called the vulva. Okay, so the area, this is a perineum, but specifically here is the vulva. Okay, so the vulva has the mons pubis, which is this area here that's directly anterior to the pubic bone. Okay, then we have the labia majora, labia minora. On this model, labia majora, labia minora, and then if you look on the other side, oh, it got happen. You see that there is a gland underneath the labia minora, sorry. That is called the vestibular bulb. It is corpus spongiosus, okay, the same one that doesn't let the erection of the man collapse the urethra. Here it's going to thicken the vaginal walls during the erection. That's like the female erection, okay? And then it coalesced into the clitoris up here at the top, okay? And the clitoris also has, where this one shows it best, corpora cavernosus, okay? Erectile tissue for the erection, and then it has a prepuce. So the labia majora comes up and makes a little hood over the clitoris. We have the same muscles that the men have in approximately the same location. Okay, so this is the bulbospongiosus. This is the ischiocavernosus. Okay, and this is going to attach, connect up there to the clitoris, and this is also going to help squeeze around and help with the or contributes to the female orgasm. Okay? And then if we look in, okay, that's not, where's my, well, we'll find it. Of course, so just the external part is called the vulva. If you open up, that up, then you'll see a couple of tubes. Who's got these tubes? This is the best one here. So you're looking in there. Any of you work with gynecology? Did you do a rotation for gyno? Yeah. So you're having to do all that kind of stuff. Okay. So in the vestibule, you have the urethral orifice, the vaginal orifice, and then laterally to the sides, you're going to have the greater vestibular or Bartholin's gland that's right here. Okay, can you see that? So that's at 4 o'clock and 8 o'clock here, and it's going to help lubricate the vagina. Okay, so is this. This is going to help. When this fills with blood, it's going to exudate into the vagina. Okay, because friction is the enemy of reproduction. Okay, like I say, ask any menopausal woman, and it's like, they don't have sex because it hurts anymore. All right, now let's go inside and let's go to the egg and see how everything's made there. Okay, so in the inside, we have the ovary. Wow. You're only half a woman. That's half a woman. I need a real woman. I can't. Okay, right there is the egg. Okay. And it's got an ovarian ligament which attaches it to the uterus. So let's find a bigger picture here. There we go, right here. Okay, so there's the egg, and there's the fimbria. This 30 is the ligament, not this. This is the uterine tube that comes in. Okay, so number 30 on this one, and then we have a suspensory ligament coming up, and it's sort of intertwined in the stuff. Okay, so I can't really tell you. If I just point there and say name this ligament, then make sure you call it the suspensory ligament. Let's see if this one is a little better. No, I still don't know. Okay, anyway. Then we have a big broad ligament, which is, who's got the better one? I guess this has a nice one here. Okay, so you can see this is the uterus. Okay, there's the ovarian ligament, there's the ovary. This big broad ligament goes all the way back, is the broad ligament. Okay, it's actually made up in 2 parts, but broad ligament is good enough. Cool? All right. This model has our stages. Okay, so we have the primordial follicle, the primary follicle, the secondary, and the vesicular should be like, see how they have it here? So that they're missing one. The vesicular follicle is the one that's about ready to burst. Okay, this has got the ovary all ready. And then the histology part is going to be all this here. So let's look at the histology. Okay, so there is a drawing. So there's all your stages that coincide with this model here, but this is the one we want to look at. Okay, so this is the mature, this is the one that's about to extrude. Okay, so this is the corpus hemorrhagicum, and then we have the corpus luteum, and then eventually the corpus albicum. So for our slide, and the slide that we have, almost pretty darn close to this right here. Okay, so around it is the theca follicula, and you can see that they look like they're traveling. One of them is closest to this area here, and then the other one's a little bit out, but you can see it's distinct from the area down there. Okay, so we got an externa and an interna. Okay, and you can see by location where the externa is and the internal is. So if I put the pointer out here, you know it's okay. It's theca externa. If it's real close to the to these cells here, then it's going to be the theca interna. Okay. Then we have the granulosa cells, which are right here. So this is the theca. There's the granulosa cells. Then the antrum is this. And then we go into well, I don't know if I have. Oh, yeah. Okay, the cumulus auriferus is that area. It looks like a stalk that goes up into it. Let me see how good we have ours here. Yeah, we have a stalk, and it's detaching just like this. So our slide has these cells that are right there, and then it's starting to detach right there. It looks almost just like this picture here. Okay, so there's your cumulus oviparous. Then we have the corona radiata, the zona pellucida, and then we have the secondary oocyte. Okay, so this is your list. If you're studying from this picture, make sure you number it according to this, A, B, C, D, and that's how you should study it, from outside to inside. And study it that way, only that way, and you'll know it forever. Okay, because that's how your brain wants to work. And I found the perfect picture. All right, so if we keep coming around, this one here is the corpus hemorrhagicum. Of course, it just means the rupture of the follicle coming out. Okay, this is the corpus luteum, and then this is the corpus albicans. Okay, so this secretes progesterone and estrogen in case of fertilization, so that can explain some changes that you might be going through because you're getting these surge of hormones. Okay, and then this is just the scar tissue that happens if you don't get impregnated. All right. Here's our fallopian tube, or your fallopian tube. I can't say ours, because I don't belong in that group. Okay, so here's the fimbria. Okay, and if you notice from these other models, the fimbria is sort of hugging the ovary. I don't know if you can see that, but you can see that fimbria is almost right on top. It really does not adhere to it. It is away, okay, because, okay, so there's extruded. This starts to flap its wings here and pull it in here, okay. So the ampulla is the fluted section, okay. Then we have the coming here. Then it narrows just before it gets into the uterus. That's called the isthmus. Okay, so we have the infundibulum, then the ampulla, then the isthmus. Cool? Okay, and usually fertilization happens about right here. And then about 5 days it travels here and it starts to implant. Cool? All right. And so the uterus, this is the fundus. So the top of the uterus. Fundus, then the body, and then we have the cervical region, okay? And the cervical region has an internal os, a cervical canal, and then an external os, okay? So in this cervical canal, we see like a little ridging on here. Here we see it too, okay? So this little ridging, this is the canal. Os, I don't know where that word comes from. I haven't looked it up, or couldn't find it anyway. But it just means an opening. And so we have an internal opening coming from the inside of the uterus and an external opening coming from the outside of the uterus, and so that's what they call that. I guess I need to look up before I comment. Do you know anything about that? Do you look at that when dilation happens and stuff like that? Do they mark like, oh, the os is so many centimeters? Okay, and as you well know, there are changes in these areas here. Okay, so we've got different sections. So the endometrium is the inner lining. Okay, so endo means within, lined with simple columnar epithelium. Okay, because it has to be very arterial, I guess is a good word. Okay, so you have to have a lot of arteries because you need a lot of blood for fertilization in here. Okay, so this layer is the one that goes through the changes. Okay, so we have stratum functionalis and then a stratum basalis. Okay, so the stratum basalis stays the same. Then every month, as the egg is developing, the uterus is also developing. Okay, so that's your functional layer. This is also what gets extruded during menstruation. Okay, then the myometrium is the muscle. Okay, so this is what propels the baby out. This is also part of the female orgasm right here. And then the parametrium is the outer connective tissue layer that is sort of continuous with the broad ligament. Okay, we talked about the broad ligament already. So the broad ligament is these in here, and then this one right here, number 29 on this model, is the round ligament, and it sort of stops here because this is like the spermatic cord, the vas deferens, and then, okay, once the DNA determines that this is going to be a female, it stops right there. And it also holds the uterus sort of in place, okay, which is important, okay, even though this will flip during arousal and stuff like that. Okay, then, then, the vagina, I'm not sure that's how you pronounce that. Okay, so the vagina is actually the opening for coitus, okay, and I don't think any model, I don't think that's supposed to be number 10. I don't think that's supposed to be the hymen, okay. So this might have a little covering on it right there that's supposed to represent the hymen. So this little covering right here, I think it's not numbered, but I'm just assuming that that's supposed to be the hymen, okay, because there's not a straight opening in there, okay. Anyway, so there's the rugae. You can see, I think this other model shows it better. So there's rugae, just like in the stomach. So there's rugae in the vagina, okay, and okay, so there's how it contributes during coitus. Okay, then the fornix is this area right here. Okay, so there's number 6 is the cervix, and it's like you took the uterus and you jammed it into the vagina, and you have this little recesses that are all the way around. That's the fornix. Okay, I got a story for you about that. When I was working at the fertility clinic, and they were taking eggs from the ovaries and freezing them, they would do the speculum thing, open that up. They'd get this needle and they'd go through the fornix here, up in that direction, and pop out an egg, take it back, and then they'd freeze it for future dates. Okay, isn't that weird? Mm-hmm. That's how they had to do with the fornix. Okay, so here you can see the fornix right around here. Okay, so this is here. The fornix comes all the way around. All right. Then the mammary glands. Okay, so this has disease processes in them. Okay, so this is trying to show the pathology that can happen in a breast. Okay, this one isn't. Okay, so I like this just for anatomy and stuff like that. And then this one is just, it doesn't show much, but you can see the nipple and the areola on there, okay? All right, so let's use this one. Okay, so we have suspensory ligaments, which in this model are right here. Okay, and they're sort of gray colored as opposed to this supposedly, I guess, fat colored. In this model here, I have no idea what it's supposed to be, okay? But, you know, suspensory ligaments hold up the breast, okay? And, you know, as we age, everything goes south, so these start to relax and you know, I have to do like this all the time. So, because I'm going south, okay? All right, then we have the lobules right here. This is where you produce the milk, okay? These are like, they're not holocrine glands, they're epicrine, yeah. So they will pinch off a little bit at the end of the cell. So when you breastfeed your babies, you're actually giving of yourself, okay? Then we have the lactiferous ducts coming in. Okay, so they're coming from the lobules, and then when you get to this area right here, there's a little swelling around there. So you have to look close when you get a chance. There's a little swelling. Let's see if this one has a big bed. Okay, so right in here, There's a little swelling, so that's your sinuses. And then, of course, we have the nipple that you come out, okay? And the nipple is generally like a pink color nulliparous, okay, dark and multiparous, so the baby can see them, okay? Because baby's extremely nearsighted. Baby can't see, you know, like there. And have you seen those videos where, you know, the newborns, once they clean them up, they put them on the baby's, the mother's chest or stomach, and it'll crawl and find the nipple. Okay, so they're very, you know that, right? That's what happened to you? Isn't that cool? That'd be a really touching moment. Okay. Oh. Okay, so it contains sebaceous glands, arousal and stimulation. So oxytocin works on this thing too. Okay, so basically nipple and areola, that's pretty easy. And that's it for the female. So let's look at the continuance. All right. So I'm missing a picture. Okay, so I told you I'd get one and then I didn't do it. Basically, this is our model. Other than some of the baby ones, because there's not a whole bunch. Okay, so you can see the stages coming here. Okay, between the fertilization. When we get to the zygote, okay, so here's the corona radiata. So this just got extruded, okay? So it goes a couple of days in the fallopian tube, and then it got impregnated, okay? And so there we have the zygote. So this is the 4-stage cell, okay? But anyway, if I tag that, this is called the zygote, okay? Then we have our germinal period. And this is a picture that I'm going to have to get for you, okay? So just use this one for the time being when I find one. And it's going to go, okay, so let's go with this thing. This is the morula, okay? The morula is a ball of cells. And then about day 5, maybe day 6 or something, a cavity forms in there. We start interjecting fluid and stuff like that, and we make this fluid here. Okay, so now it's a hollow ball of cells with massive cells at one side. Okay, so there is the morula. I need to get that picture right here because it needs to go right there. And this is the blastocyst. Okay, and it's going to have the trophoblast and the inner mast cells. Okay, so this is what gets tagged. Okay, on a picture that goes in between here. Okay, then we have this stage here, okay, which is our bilaminar disc. This is the trophoblast, but it's do I have anything that I can tag for that? Okay, so I can't tag this in well, I guess I'll have to use this picture if I want to do that. But these two things right here are here and there, okay? And they're real important too, okay? So there is this one here, okay? If you turn it this way, that's this one here, okay? So this is the bilaminar disc. This is what this is called. Okay, so 1, 2, bilaminar disc, okay? And we have a hypoblast and an epiblast. Okay, so the hyperblast is going to be, I mean, when you study this picture here, it'll be this is the orientation according to the picture when you do like this. Okay, so 27 would be the hypoblast, and then the epiblast doesn't have a number. Okay, but that's the yolk sac. Okay, then we have these two things here. Of course, this is the amniotic cavity right here. Okay, so we have a syncytiotrophoblast and a cytotrophoblast. Okay, so this is going to become the chorion, and then this is going to help burrow into the uterus. Okay, and it's going to burrow this whole thing is going to burrow all the way in, and it's going to be surrounded by all that blood. Okay, so that it can start giving the nutrition that it needs. gastrulation, we won't be doing primitive streak. But we are going to be doing ectoderm, mesoderm, and endoderm. Okay, so here's our third layer that we're missing. Okay, which is this one here. Okay, in fact, it's that picture pulled out. Okay, so it goes right here. Okay, so before and after the bilaminar disc, we should have one here, we should have one there, and I'll get a picture that will show that. Okay, and gastrulation means the formation of the 3 layers. Okay, now that we have these 3 layers made, we have an ectoderm, a mesoderm, and now an endoderm, the yellow, down here. Okay, so that, now we have the beginnings of everything that we need. So we got the skin and nerves here. Then we have the mesoderm, muscle, bones, kidneys, gonads, etc. And then the endoderm is going to be your gut, the mucosa, digestive, respiratory, urinary tract. Everything that we just learned. All that comes from just this area here. Cool? All right, so this ball is going to start extruding like this. Then it's going to fold on itself, and we're going to get to this picture right here. Okay, so you can see there's that. Okay, these is starting to become all this stuff here. This eventually is going to get absorbed in and become the gut tract. Okay, so there's the amnion. Okay, so this right here, 32 and 30, is the growing embryo, right here. So 31 here is the amnion. Then 33 over here is the yolk sac. Okay, so when you study this, if you take a picture of this model, you have to sort of turn on its side so that it matches this picture here. Okay, so there's the amnion. This is the yolk sac, and then 36 is this part here, which is called the allantos. Okay, which is eventually going to become the umbilical cord. Cool. All right. Then we look at the placenta. Okay, the placenta has 2 parts, one made by the baby, one made by the mother. And where is that little baby? Look at you, you cute little baby. Oh, I call all girls girl dogs because I'm so used to saying that. When I see Bella, I have to fight not to call her a girl dog. All right, so here we go. Remember, here's baby. Okay, we're looking at a different stage here. Okay, so this is the placenta here. And you can see the venous side is the baby side, and the pitted side is the mother side. Okay, and that's because the villi there, and then the spaces that the mom has on her side. Okay, so chorionic villi, finger-like projections. Let's see if I can find a better one. Will you hold my baby? Okay, so here's the villi. So the villi obviously are red and blue, and then the gray is the intervillous space from the mother. Okay, so that's part of the decidua basalis. Okay, the decidua is the maternal portion, the chorion is the fetal portion. Okay, so if I ask you to name this section or name this here, you'd say chorion. Okay, and then if I ask you to name the other part, depending on where I tag it, okay, so the decidua basalis is just this area right here, where we have contact with the baby, the villus. Okay. Then the other side that surrounds that is the capsularis, area of placenta that covers the embryo. Cool? All right, you can take care of that baby. And then the arteries and veins. Okay, so what do we need to know about the arteries and veins? The arteries are going to be actually blue colored in the baby. and the veins are going to be red colored. Okay, because it follows the rule. And we're talking about the baby. So in this case, the arteries are going to take the waste product away because the baby is not going to do all the exchange in his lungs. His lungs aren't developed. Mother's going to do all that up there in the intervillous space in the villi. Okay, so mom's going to dump a load of blood into the intervillous space, and then baby is going to extrude the waste and then absorb the nutrients. Okay, so in this case, arteries are going to be blue and veins are going to be red because it's going to take blood to the heart. Cool? Do we have another one that could show that better? Where's my other little baby? Did you lose my baby? Oh, there it is. Okay, I was about to cry. Okay, so here we have. Okay, so remember, the red is the artery. The red is the vein, and the blue is the artery on this one. It's opposite the convention that we talked about. Cool? All right, that's it. This is a very doable test.